Hello students, welcome to the lectures of hematology, and we will see about the blood. What the blood consists of? Blood consists of large amount of plasma and the cellular components. What the plasma consists of? Plasma consists of the plasma proteins, albumin, globulin and fibrinogen. Then we have got about some electrolytes and the water part. And when it comes to the cellular part, the three important cells which are there, RBC, WBC and platelets. So in today's lecture we will see about mainly the red blood cells or the RBC. What are RBCs? RBCs, we can say they are the bags of hemoglobin. They have hemoglobin incorporated in them. Now first, what is the importance of hemoglobin? Why the hemoglobin should be there in the blood? Remember that hemoglobin increases the oxygen carrying capacity of the blood nearly 70 times. Without hemoglobin, very less amount of the oxygen that will be carried by the blood. So what is the most important function of the hemoglobin? It will increase the oxygen carrying capacity of the blood. Next, the hemoglobin will also act as a good buffer. Now when this is required? When lot of carbon dioxide from the tissues is there transported into the blood. Now carbon dioxide will generally try to decrease the pH. It will try to increase the acidity. In that case, what will happen now? The hemoglobin will take up most of the H plus ions and then it will act as excellent buffer. So that is another function of the hemoglobin. Now as I told you that the hemoglobin should be packed in the RBC. Why hemoglobin cannot be free in the blood? What will happen if it is free in the blood? If it is free in the blood, number of things that can change. First and important thing, it will be filtered. It will be filtered at the capillaries. It can go into the tissues. Again, it has to be returned back to the blood. Next important thing, it will also be filtered at the glomerulus. So once it is filtered in the glomerulus, it will go into the nephron, and once it goes into the nephron will try to reabsorb it back, but whatever has not been reabsorbed, that will be excreted. So large amount of hemoglobin that will be excreted. Not only that, if the hemoglobin is free inside the blood, then it is also going to increase the viscosity of the blood. So because of all these things, the hemoglobin should be packed in the RBC. So that's why the RBCs are there, which has got the hemoglobin incorporated in that. One more important function of the RBC is that the RBC membrane, they have got one important enzyme, carbonic anhydrase. Now just recall, what is the function of carbonic anhydrase? Carbonic anhydrase will catalyze the reaction between CO2 and H2O. And what will be formed? H2CO3, and which can be dissociated into H plus and SCO3 minus. So because of this, now this reaction is taking place. Because of this reaction is very fastened up by carbonic anhydrase. And because of that, lot of amount of CO2 can be easily taken up by the blood at the tissues. So the CO2 transport, for the efficient CO2 transport, carbonic anhydrase is required. And who has got that carbonic anhydrase? The RBC cell membrane. So then, what are the functions of RBC? RBC, since it has got the hemoglobin, so oxygen carrying capacity is increased 70 times. Then next, the hemoglobin can act as a good buffer, and because of the carbonic anhydrase, the CO2 carrying capacity of the blood is also going to be increased. Right? Now, we'll go for the size and shape of the RBC. If you have seen about the RBC, what is the typical shape? It is the biconcave disc shape. So this is just a two-dimensional structure. If I want to have the three-dimensional structure, the biconcave and the disc shape structure. Now the diameter of the RBC is 7.8 micrometer. The thickness, that is at the thickest point at the periphery, is 2.5 micrometer, and in the center where the thin part is there, it is 1 micrometer. Now the question is that why this peculiar shape? Why it's not spherical? The number of advantages are there. Mainly the cell membrane proteins, they try to maintain this shape. Now what are the advantages of this shape? It has got the pliability. Pliability means what? The RBC, it has to squeeze through the capillaries. It has to fold, it has to mold. And because of that, the squeezing and molding, this is the property which is called as pliability without breaking up the cell membrane. So the RBC can pass through the capillaries, squeezing through. This is the diapedis, we can call it as the squeezing through. So because of that, this next important advantage is that RBC is generally faced with the hypotonic. If you drink large amount of water, plain water, distilled water, what will happen? Your blood will become hypotonic for some time. In that case, what will happen? If the RBC is kept in a hypotonic solution, what is going to happen? Naturally, the endosmosis will take place. That means the fluid from the surrounding spaces will enter inside the cell. And in that case, what will happen? The cell size will increase. If it would have been spherical now, what would have happened? Then the RBC will not be able to resist any change in the osmolarity of the blood. So since it's having a biconcave spherical shape, remember this biconcave spherical shape is just opposite to that of sphere. Now if you recall, why a drop of water which is from the tap, it will try to take a spherical shape? It is because of the surface tension. Yes, recall your knowledge. And what the surface tension will try to do? Because of the surface tension, the drop will have to accumulate least surface area. So sphere is the one which is having the maximum volume in the least surface area. This is the one which is having the more surface area in that is the volume which is having the maximum surface area. Now what is the importance of more surface area? Because of the more surface area, there is exchange of the gases that can be facilitated. So rapid exchange of the O2, rapid exchange of the CO2 that can be facilitated. So this is one more important function of that. Why there is a biconcave shape? It provides more surface area. Because of that, what is going to happen? There is a more surface area for the exchange of the gases. Right? So because of that, it gives the RBC has got the more stability. It will oppose the fragility, and it is also going to have the more surface area. What is the average volume of the RBC? 90 to 95 cubic micrometer. How much is the normal count of RBC? Generally we say it is 5 millions per cubic millimeter. That is, it is more in male compared to that in the male we can say is 5.2 millions per cubic millimeter, and in female it is 4.7 millions per cubic millimeter. Why RBC count is more in male? This is a commonly asked question. Why the RBC count is more in male? It is because of the testosterone or the androgens which are more. The androgens will have the more metabolic effect. Ultimately, it is going to increase the more muscle mass, and also it is going to have the more anabolic effect, and because of that, the RBC count is also going to be more. So that's the important thing. Now, how much is the quantity of the hemoglobin which the RBC can be there? Maximum amount of the hemoglobin that can be there in about 100 milliliters of RBC is 34 grams. That much is the maximum amount of this. Concentration of hemoglobin, when the hematocrit, that is the percentage, cellular percentage of the blood, that is if it is normal range is 40 to 45%. So if the hematocrit is normal, in that case in male the concentration of the hemoglobin will be 15 gram. The range can be 14 to 18 gram. 15 gram percentage, or we can milliliter per 100 milliliters of the blood. And in female it is 14 grams. Again the range can be from 12 to 16 grams. So that is the per 100 milliliters of the blood. So that much is the amount of the hemoglobin that should be present. And one more important thing you should remember, each gram of pure hemoglobin is capable of combining with 1.34 ml of oxygen. So this can be asked as a multiple choice question, right? So each gram of pure hemoglobin is capable of combining with 1.34 ml of oxygen, right? Next, going to the erythropoiesis or genesis of RBC. Now, what are the sites where the RBCs are produced? In the fetal life, the RBCs are produced in yolk sac. They can be produced in spleen. Then liver will also take part in the genesis of RBC, or it can be in the lymphoid tissue. But when the baby is born, the most of these organs, the function of most of these organs will be taken up by the bone marrow. The bone marrow, that is the red bone marrow, and slowly when the adulthood comes, the most of the red bone marrow is again converted into the yellow bone marrow, and what is left is generally the bone marrow of the membranous or the membranous bones, such as vertebra. It can be in the skull, it can be in the ilium, or it can be in the sternum. So these are the places where in the adults the RBCs they are produced more. These are the sites of synthesis of the RBC. And the hematopoiesis as such is controlled by the growth inducers as well as the differentiation inducers. Now what do you mean by hematopoiesis as such is controlled by growth inducers and the differentiation inducers. What do you mean by growth inducers? They will cause the growth. Differentiation means what? We will see that the pluripotent hemopoietic stem cell, again it should be differentiated into different forms so that it can form various WBC, it can form platelets, it can form RBC. So this function is again done by the differentiation inducers which are there. And this growth inducers and differentiation inducers, they are in turn controlled by a number of other factors. To give you an example, in case of erythropoiesis, whereas RBC production is there, this erythropoietin is the main hormone which is controlling this growth inducers and the differentiation inducers, and in turn it is going to cause the production of the RBC. Now coming to the hematopoiesis. So if you look at this flow chart, we have got the important cell which is present in the bone marrow. This is called as pluripotent hemopoietic stem cell. The name itself suggests pluripotent. That means it has got the potential to form number of different types of the hemopoietic stem cells, right? So pluripotent hemopoietic stem cells, it will divide into mainly three forms, that is three cells. It will divide into pluripotent hemopoietic stem cells which will act as a reservoir. Then it will divide into colony forming unit spleen, and then it will divide into lymphoid stem cell. Now this lymphoid stem cell, they can divide into T lymphocytes and the B lymphocytes. Whereas the colony forming unit spleen, it will divide into colony forming unit blast, which are again divided into colony forming unit erythroblast, and which will ultimately form the red blood cells. So we will get our red blood cells from the colony forming unit blast and the erythroblast. Whereas the colony forming unit spleen can again be divided into colony forming unit granulocyte and monocytes. That means they are going to form the granulocytes such as neutrophil, eosinophils and basophils. And we have got colony forming unit monocyte. So that means it is going to form monocytes, and the monocytes, when they enter inside the tissues, we will see in detail about this, but this is very important thing. Monocytes, when they enter into the tissues, they convert into the large macrophages. So macrophages are mainly derived from the monocytes, right? And then we have got this the colony forming unit megakaryocyte, and ultimately megakaryocyte, it is a big large cell, and from the megakaryocyte that will be formed, and the pinching of the megakaryocytes, the small parts of that are nothing but the platelets. So ultimately the platelets will be formed there. So this is about the overall hematopoiesis. Now our today's lecture we will be seeing about the erythropoiesis. So as we have seen from the erythropoiesis, from the erythroblast, now what are the changes that are taking place? Blast means what? A premature cell that can be called as a blast cells. A premature large, generally it will be a large cell, it will be having a big nucleus. So these are called as the blast cells. Okay, right? So we have got, what are the steps in the erythropoiesis now? We have got pro-erythroblast, that is the number one cells which can be identified. Then according to the staining pattern, generally we call it as the basophil erythroblast. That means it's mainly having the bluish color, that is on the staining, right? Then we have got is the polychromatophil erythroblast. Poly means what? That is the number of chromatophils, that is colors that can be seen. It's because of the more and more amount of the hemoglobin is now being incorporated, right? So the eosinic stains that will be there, right? So we have got the polychromatophil erythroblast, and then it is divided into orthochromatic erythroblast and ultimately reticulocyte. So this is an important thing, the reticulocyte cell and then the erythrocyte. So these are the stages of the genesis of the RBC, right? Now what are the changes that are taking place in the RBCs? What are when the erythropoiesis is taking place? Important thing you have to remember, as I have told you, they are the growth inducers and the differentiation inducers. So ultimately what they are going to cause? Differentiation. Differentiation means what? From one proerythroblast, number of basophil erythroblasts that will be formed. So the number of cells they are going to increase. Next, as I have told you, blast cells are the bigger cells which are having the nucleus, and what will happen now? The size of the cell is going to become smaller. So that is the second change in the maturation, we can say, or the genesis of RBC. Next, most important thing, the hemoglobin synthesis will start, incorporation of hemoglobin. And if you remember the structure of RBC now, the RBC does not have nucleus, it does not have mini cell organelles, it does not have mitochondria, it does not have endoplasmic reticulum. Where they have gone? They have been extruded out. So ultimately the nucleus will become pycnotic, it will become small, it will be extruded out, right? So endoplasmic reticulum, Golgi apparatus, they have been extruded out. The remnants of these cell organelles that can be seen in some stages such as reticulocyte. So what is reticulocyte actually? The reticulocyte, if I want to draw a structure, I will see some reticulum. This I can call it as the reticulum. What is this reticulum actually? These are the remnants of the cell organelles. And just as endoplasmic reticulum, mitochondria. So reticulocyte. Reticulocyte generally count is 1 to 4% because the reticulocytes will be released and ultimately the maturation, some maturation will also take place in the blood. The reticulocyte count is 1 to 4%, but the reticulocyte count also has got the importance. In a person, if you see that the reticulocyte count has increased, what does it mean? What the inference you can draw from that? That the person has recently suffered from hypoxia. And because of that, the erythropoiesis rate has been increased, right? So that's the inference you can draw it from the increase in the reticulocyte count. Now we will see what is the main stimulus. Who regulates the number of RBC in the blood? You might be thinking that is the number of RBC. If 5.5 millions per cubic millimeter of blood, if it is more than increased, then it will cause feedback inhibition, or it is related to the hemoglobin concentration. But it's not the case here. The important thing you have to remember is that what is the function of hemoglobin, what is the function of RBC? To carry oxygen. If the oxygen carrying capacity of the blood is decreased, then this will have the effect over the bone marrow and ultimately it is going to cause production of more RBC. Remember, even if the hemoglobin content in each RBC it has decreased, the RBC count might be normal. But if the hemoglobin concentration has decreased, so what will happen to the oxygen carrying capacity? Oxygen carrying capacity will be decreased, and in that case there will be more tissue hypoxia, and tissue hypoxia will stimulate the bone marrow and ultimately it is going to cause more production of RBC. Same thing can happen in case of a hemorrhage also, where the hemoglobin percentage, that is hemoglobin concentration is normal, but the oxygen carrying capacity of the blood is decreased because of the absolute count of RBC is decreased. Or if the person goes to high altitude, in high altitude the hemoglobin percentage is normal, the RBC count is normal, but since the partial pressure of the oxygen in the atmosphere is less, the person is receiving less amount of oxygen. In that case the oxygen carrying capacity has decreased. So in this case also what will happen now? The person will suffer from hypoxia and the RBC count will be increased, and that condition is called as polycythemia. Now, how does the signal from that is the tissue hypoxia, how does the signal goes to the bone marrow? Tissue hypoxia, that means the hypoxia will also be there to the kidney and the liver as such. These two organs mainly, they synthesize a factor which is called as erythropoietin. Mainly it's the kidney which is going to synthesize 90% of that. The kidney will synthesize erythropoietin. So if less oxygen is reaching to the kidney will synthesize the erythropoietin, and that erythropoietin will act over the bone marrow and ultimately it is going to cause the more synthesis of the RBC. If, as you can see in this flowchart now, Hematopoietic stem cells, proerythroblast, RBC, and ultimately what is the function of the RBC? To cause tissue oxygenation. If this is decreased, such as conditions low blood volume, anemia, low hemoglobin, or any of the pulmonary disease also, if the tissue oxygenation decreases, in that case what will be there? Erythropoietin will be released from the kidney, and ultimately this erythropoietin will act over the hematopoietic stem cells to cause more genesis of the proerythroblast. So the renal hypoxia causes what? Increased level of hypoxia inducible factor 1, which act as a transcription factor for the erythropoietin gene. More erythropoietin that will be synthesized, and ultimately it will act over the bone marrow. Now whenever the synthesis of the RBC is taking place, as I have told you, there is a cell division. From one proerythroblast, number of RBCs they are being formed. And for the cell division, what is required? Mitosis. Mitosis, what is required? Nitrogenous bases such as adenine, guanine, thymine, cytosine. Now, for the synthesis of thymine, one important, that is vitamin B12, folic acid, these are required. And vitamin B12 deficiency, if it is there, what will happen in that case? The cells will not be able to divide. They will remain as a blast cells or the premature cells. They will remain as blast cells. And since there is a need of the body, these blast cells will be released into the blood. So if you look at the peripheral smear now, what you will see? That the RBC cell size has increased. The nucleus will be there in them. This will be called as erythroblast. They will be seen. And this is classically seen in vitamin B12 deficiency. So this can be a multiple choice question asked. What is the exact role of vitamin B12 in the RBC production? So what you should write? They are required for the vitamin B12 is required for the production of the thymidine triphosphate, again which is required for the mitosis. Vitamin B12 absorption, if you recall your gastrointestinal system knowledge, what is required for the vitamin B12 absorption? Intrinsic factor from the oxygenating glands. In atrophic gastritis, when the cells of the stomach they are destroyed, intrinsic factor is not being produced. And if the intrinsic factor is not produced, what will happen in that case? Vitamin B12 will not be absorbed. And in this condition, that will be called as pernicious anemia. So what is pernicious anemia? Generally autoimmune atrophic, that is destruction of the gastric mucosa. Because of that, the intrinsic factor will not be produced. And ultimately, no vitamin B12 that is absorbed. No formation of that is less formation of the thymidine triphosphate. And ultimately the proerythroblast that will be released into the blood, and ultimately what you will have is the megaloblastic anemia. So that is because the now coming to the hemoglobin synthesis. Hemoglobin is incorporated in all the stages of the that is the synthesis of the RBC. And how the hemoglobin is synthesized? So if you look at into this charts, 2 succinyl-CoA will combine with the 2 glycine amino acids, then what is formed is the pyrrole ring. This 4 pyrrole ring, ultimately it is going to form protoporphyrin 9. Protoporphyrin 9 will combine with iron to form heme molecule. And this heme molecule will combine with the polypeptide. In case if the hemoglobin A has to be formed, then it should be alpha and beta chains. So ultimately, heme will combine with the polypeptide to form heme chain. Either it can be alpha chain or it can be beta chain. And now 2 alpha chains and the 2 beta chains, ultimately it is going to form the hemoglobin A or adult hemoglobin. There are different forms of hemoglobin. Hemoglobin F is also there, that is fetal hemoglobin. That has different characteristics. It has got the more O2 affinity. So it depends now, that's the different types of chains they are produced. They are present in that. One has to remember that each molecule of hemoglobin can combine with 4 molecules. That means the 8 atoms of the oxygen. And this is very important point, that the combination of the hemoglobin with oxygen is a reversible combination by the that is called as the coordination bonds. Reversible combination which can be easily, again the oxygen can be released by the O2, right? Next, coming to the iron metabolism. Iron is very important for the synthesis of the hemoglobin. So iron is mainly stored in the ferritin form, that is in the tissues, mainly ferritin. If you look at overall the iron metabolism, just look at this chart. What you can see here, the iron which is there in the body, it can be in the tissues. Either it can be in the storage form, that is ferritin. Either it can be in the hemosiderin, either heme or some enzymes. And so which are the some, such as iron is also present in myoglobin. It can be present in cytochrome oxidase, peroxidase, catalase. So apart from hemoglobin, these are the other enzymes and some molecules which also contains iron. Now this is which can be converted into free iron from that, and this free iron will go to the plasma, and in the plasma it will be carried by the transferrin. This is the protein which carries the iron. If it is not having the iron combined with it, that will be called as apotransferrin. And remember, this apotransferrin has got a very important role in causing absorption of iron from the intestine. So the amount of apotransferrin will determine how much amount of the iron is absorbed from the intestine. And what determines the amount of apotransferrin? It is if it is not bound with the iron. So if it is bound with the iron, it will be converted into transferrin, right? And from the plasma, it can be excreted some amount of the iron that can be excreted 0.6 milligram per day. Or now this can be used for the synthesis of hemoglobin, and when it is used for synthesis of hemoglobin, there can be loss even to some of the bleeding disorders, or it can be in the cases of a woman, it can be lost to the menses. Blood loss to the menses is 0.7 milligram of iron per day in menses. That is this. And what will happen when the RBCs get age old? The normal lifespan of RBC is 120 days. So once they get age old, after they get destroyed when they pass through the small capillaries. Okay, they get hemolyzed when they pass through the capillaries. And the hemoglobin is then taken up by the macrophages. And the macrophages will cause the breakdown of, it will separate heme from the globin part, and ultimately the bilirubin synthesis that also will be there, and from the heme the iron will be separated. The iron part which is there, ultimately it will be used for the synthesis of the more and more amount of the hemoglobin, right? So it is mainly the function of the macrophages. Degrading hemoglobin, okay, the porphyrin part which will be converted into bilirubin, and whereas the iron part that will be converted, that is the free iron which can be used again. As I told you, the RBCs when they become age old, the metabolic system becomes very weak, and not only that, the pliability of the membrane becomes very less now. Since it does not have the nucleus, it does not have the mitochondria, it is not able to maintain that metabolism for a very long time. So the normal lifespan of RBC is 120 days. And when it is passing through the narrow capillaries now, especially for the spleen will act as a graveyard. So they will get ruptured, right? And ultimately the hemoglobin will be taken up by the reticuloendothelial cells, and the porphyrin part will be converted into bilirubin, right? So with this we come to the end of this lecture. Thank you.